DNA replication depends entirely on that base pairing, so A's to T's and C's to G's. And when we do DNA replication, what we have to look for is the DNA strands to separate. We've got to get them apart, so breaking hydrogen bonds. And then we look at each strand as a pattern for the new DNA. So it's something called the Semiconservative Replication model. When we take our original strand of DNA, we're going to split it apart and use each strand from the old DNA as a template or as a model for the new strand. And the way we do that is if we have a T here, we're going to add an A for the new strand. Well, if we have an A here, we add a T for the new strand. And when we end up putting these back together, you notice these two strands should be identical based on the way we've done the semi-conservative replication. Here it is in a more realistic sense. We don't have little tiny straight pieces of DNA. It's in that helix form. And we are not going to actually pull the entire DNA strand apart. You have to keep in mind that some of your chromosomes that we're trying to replicate are hundreds and hundreds of thousands and upwards of millions of base pairs long. We can't just pull those all apart. They're just going to stick back together like little magnets. So we have to actually do this in a very systematic way that we're going to show you. There are 7 major steps to DNA replication. And the first step is to actually get in there and get the strand pulled apart enough to do replication. So we form what we call a replication bubble. So helicase is going to unzip the DNA. Primase is its partner in this, and we'll look at what primase does in a little while. You know that ACE and ACE are enzymes, so you know that they're going to catalyze or cause something to occur. As helicase is unwinding this DNA, it's only going to do it in very small pieces. So as it's unzipping and unwinding, we form something we call a replication bubble. These replication bubbles start to unwind and allow DNA to be replicated in 2 directions at the same time. This speeds replication because remember, in your cells, in interphase, in your cells, in S or synthesis, we look at DNA replication occurring in about an hour. So how does this go so fast? It's because of these replication bubbles. So the replication bubbles are going to allow us to start replication in multiple places along the DNA at the same time, speeding up this process. We look at DNA replication occurring in what we call the 5' to 3' direction. These prime numbers are based on the sugars, the carbon positions on the sugars. I'm not super concerned about you thinking about the reason why it's named this, but what I want you to think about is that when we look at replication on the 3' to 5' template, it goes very fast. It's what we call continuous. The other one, the 5' to 3', goes in what we call a discontinuous region. And this is why. When we look at the 5' end, we've got a phosphate. The 3' end, we've got the OH from the sugar. So the 5' comes from its orientation of the phosphate off of the 5 carbon, and the 3' in the other direction, so the 3' here in the OH from the sugar. And what we look at is, this is something known as an anti-parallel design. It means that it's running in opposite directions with 5' at one end and 5' at the opposite end on the other strand. This allows the DNA to actually have that twist or spiral to it. But what it means for our DNA replication is that if our DNA polymerases that are going to be responsible for adding nucleotides to our replication process, if they are able to attach to the 3' end of this strand, they're able to then attach a phosphate, a sugar, and a nucleotide and get going with replication very quickly. However, if we open up this other side where we have a 5' end at the end, this is a phosphate, remember, on the end, all the nucleotide can add and all the polymerase can add is another phosphate. Well, we don't want a phosphate side by side, so we actually have to go back and do what we call priming the strand. So we're actually going to add primers to this strand to give a starting point for the DNA polymerase. So two big things we're going to need are the DNA ligase. This is the DNA glue. This is what is going to hold everything together and make sure that everything is aligned properly. The DNA polymerase adds nucleotides. It also does other things. There are several forms of polymerase. And these several forms of polymerase, one will add nucleotides, one is going to do proofreading. So literally, like if you're proofreading a paper, making sure the letters are in the correct order. And then it's actually going to do some correction if the proofreading shows error. So in this process, we're doing DNA replication, but we're also doing DNA repair at the same time. The goal for DNA replication, of course, is to be perfect. It doesn't quite make it. So when we look at the DNA replication process, it's going to be near perfect. And when it's not, we're going to rely on DNA repair mechanisms with the DNA polymerase and the DNA ligase to come in and help correct any problems.